Hi, my name is Trinity, and I will be presenting with Emily today. Our project for this year is cGEM, which is about engineering potatoes to express antimicrobial peptides as a solution to problems concerning agriculture and human health. We are the University of Lethbridge cGEM team, located in Alberta, Canada. In our team's location, agriculture is one of the most prevalent industries. So, we have decided to focus on an agricultural-based project to suit our geographical needs. We will begin with a brief introduction to potatoes, the focus of our project. When choosing to use potatoes for our project, the large production and consumption rates played a large role. Since the potato industry impacts many people, whether as producers or consumers, we wanted to know more about the statistics regarding potato production and consumption rates here in Canada. In 2016, 4,770,521 tons of potatoes were produced in Canada. In 2011, 78.37 kilograms of potatoes were consumed on average per capita in Canada. Canada also has a large role in the export industry for potatoes. In the 2015-2016 fiscal year, $1,597,265,233 worth of potatoes and potato seed products were exported out of Canada. In order to help design a project that could be truly useful to the potato industry, we started by taking a look at how potatoes are produced. The potato production line begins with seed producers. They propagate the seeds and go through many trials to try and ensure the seed varieties they sell are disease-free. After speaking to farmers, we have learned that the seed producers chosen are often selected by mere chance and then maintained only if they continually provide disease-free and aesthetically appealing varieties. Next, the farmers take the seeds and grow the potatoes. Some produce locally, but many supply to larger companies. The variety, appearance, size, shape, and method of preserving the potatoes are strictly regulated by the companies they provide for. After the potatoes are grown, they are put into storage until the companies can use them. This time frame can span anywhere from 2 weeks to 10 months. Finally, the manufacturing companies process the potatoes into the desired form, whether it be hash browns, fries, chips, or whole potatoes. Those products are then shipped to other countries or consumed locally by Canadian residents. Looking at this production line, we wanted to think of the main problems each of these sections encountered to see which issues we could address. In terms of seed production, crop production, and storage, the primary concern is disease and damage, particularly during storage. After speaking with farmers and doing research, we decided to focus on one of the most prevalent potato pathogens, Fusarium graminearum, a fungal pathogen which causes dry rot. As many Canadian potatoes are exported, engineering potatoes to help produce pharmaceuticals could also benefit consumers in developing nations. Treatment of a disease is often expensive, so the ability to have easy access prevention methods for cheaper would be extremely beneficial. For consumers within Canada and around the world, we would like to increase their nutritional value to contribute to the overall health of the population. Looking at these three very different problems, we tried to identify a way we could develop one method to address all of them. To do this, we planned to engineer potatoes to express antimicrobial peptides, or AMPs, with desired effects. This way, all the problems can be addressed by developing one methodology and simply altering the AMP produced. Before we begin, I would like to explain what AMPs are. Antimicrobial peptides, or AMPs, are peptides produced by a variety of organisms as a defense mechanism against parasites such as bacteria, fungi, yeasts, or viruses. Since many AMPs act against a wide range of parasites existing in nature, they have a lot of potential for use in synthetic biology, especially in relation to agriculture and human health. Many AMPs also exist naturally in crops that are regularly consumed, so the toxicity risks to humans would be relatively low. To narrow our focus in regards to these three issues, we have developed three facets to our project to focus on. The first problem we would like to address is that fusarium infections causing dry rot are found in most potato seeds. This causes large monetary and supply losses to farmers and commercial businesses. Expression of AMPs effective against fusarium in potatoes would provide a biological control of fusarium within the potato plant. Using a topical application of AMPs to combat fusarium would provide a similar effect without genetic modification of the potatoes. This system would improve the current methods for dealing with fusarium since, after discussions with local farmers, it was revealed the current methods are extremely time-consuming. The second problem we would like to address is that pharmaceuticals are not affordable in many developing nations. Expression of medical-grade AMPs in common crops allow for production of inexpensive pharmaceuticals for developing areas. For our project, we are focusing on the treatment of sleeping sickness caused by the Trypanosoma species in sub-Saharan Africa using the bovine AMP BMAP 18. The decision to focus on this particular disease came from research done with the Uvo Shento lab, which we are collaborating with and who have begun research on this particular disease already. Finally, the third problem we are exploring a solution for is that in many cases, treatment of a disease is done retroactively. Rather than being prevented, the disease is treated only when it has already become a problem. Our solution is that by expressing AMPs with known health benefits in food that is consumed regularly, we can take a proactive approach. The AMPs will help to increase immunity to potential health threats so diseases are less likely to occur. This doubles as a health benefit to healthcare systems, which would be less likely to be overwhelmed when outbreaks occur. Due to restrictions resulting from the COVID-19 pandemic, the first few months of our project for the first year were spent virtually designing constructs and doing extensive literature reviews to better understand the basis and directions of our project. Since we still do not have wet lab access until mid-September, we have decided to elongate our project into 2 years to give us the time needed to properly complete it. Moving into the next couple of months, we will begin to transform E. coli with our constructs in order to test AMP expression. Once we have successfully developed a method where this can be done, and the AMPs can be produced by the E. coli, they will be purified and the peptide's properties will be tested. Next, we will follow the same steps to transform potatoes with our constructs and purify the AMPs produced by the potatoes. Once the AMPs can be successfully produced in potatoes, we will begin by testing the efficacy of the peptides to ensure that they are able to do what we have intended. Since the information on the peptides came from literature reviews, we will also test them on our own to ensure we can provide the same results. Next, we will test several biological components, such as ensuring the peptides won't denature during the digestive process. Within our team, we have 3 lab teams which are researching different aspects of our project. The first one, being the plant team, is working on growing, preparing, and transforming potato plants with the plasmids developed by the construct design team. The plasmid of interest will be introduced to the potato plant by agrobacterium-mediated transformation. Agrobacteria uses a tumor-inducing plasmid to transfer DNA into plants. The transformed plants will be tested for expression, and the best plants will then be propagated and grown.
The construct design team has been investigating parts and assembly methods for designing plasmids for AMP expression in both E. coli and potatoes. The current focus is on designing constructs for the expression of BMAP 18, an AMP that acts against the sleeping sickness parasite Trypanosoma, as well as AMPs that act against Fusarium graminearum and other fungal pathogens of potatoes. The E. coli constructs serve 2 purposes. First of all, they allow us to produce AMPs more quickly and easily than in plants for preliminary testing purposes. Secondly, they allow us to produce AMPs for topical application on crops, which is a method we are also exploring as an alternative to engineering the potatoes themselves for farmers who may be opposed to the idea of GMO crops. Our E. coli constructs were designed using BioBrick assembly on the standard PSB 1C3 backbone. We have chosen to use the IPTG-inducible optimized PTAC promoter in these constructs, as it has been previously optimized for use in E. coli. It also allows us to control the expression of our peptide, therefore allowing the E. coli cultures or colonies to grow to a sufficient size before they divert energy into the expression of the AMP. All of our E. coli constructs contain the standard RBS B0034 and the standard double terminator B0015. We have so far designed E. coli constructs for BMAP18, as well as the antifungal AMPs peptide G2, snakin-1, snakin-2, TUAMP2, cGEM17, and cGEM100. These AMPs were chosen because they are not toxic to humans, and peptide G2, snakin-1, and snakin-2 actually come from potatoes. cGEM17 and cGEM100 are also thought to have probiotic effects. Our potato constructs were designed using Golden Gate assembly on the standard Golden Gate Level 1 iGEM backbone P10500. The promoter we are using is the strong constitutive promoter CAMV35S from the cauliflower mosaic virus, which has been shown to be particularly effective in plants. All of the potato constructs contain the Kozak sequence to facilitate the initiation of protein translation. We have designed plasmids for each AMP with the CAMV35S terminator as well as with the NOS terminator, as both have been shown to be effective terminators for use in plants. As with the E. coli constructs, we have designed potato constructs for BMAP18, as well as the antifungal AMPs peptide G2, snakin-1, snakin-2, TUAMP2, cGEM17, and cGEM100. We are currently in the process of adding tDNA repeat borders to either side of our constructs, as well as an agrobacterium origin of replication so that they can be used in agrobacterium-mediated transformation. The peptide purification team prepares, extracts, and characterizes the peptides being expressed in both E. coli and potatoes. Our strategy involves first identifying a variety of potential antimicrobial peptides. The peptides we chose to research came from a range of different organisms. Some were native peptides to the potato, but some came from other organisms, such as BMAP18 from cows. Secondly, we looked at the properties of these peptides by reading through different research papers that examined each peptide. This also allowed us to assess whether the peptides could be harmful to humans, and such peptides were eliminated. After this, our third task is to develop an optimal purification method based on our research of current methods and protocols for the preparation, extraction, and characterization of our chosen peptides. Once the potatoes are grown and ready for experimentation, we can test out the methods we developed. The diagram illustrates the purification process of reverse phase high-performance liquid chromatography. This method involves separating particles based on their reactions with water molecules so that compounds with high hydrophobicity come out later on the chromatogram, and those with low hydrophobicity come out earlier. This is the primary method we are planning to use to purify our proteins, with minor adaptations to be made later once lab access continues to open up. The modeling team is looking into developing molecular dynamics simulations based on canonical peptides found in literature. This first involves locating AMPs of interest that have a crystal structure and a site of interaction. Although not all of our peptides have identified crystal structures, for the current iGEM season we are focusing on those that do to allow for more seamless simulations and effective training for new members to the subgroup. Once we identified the available crystal structure, we began a literature review to see where these AMPs react naturally. In order to run a simulation, we require the interaction site of the peptide, and also require the molecules they interact with. Although no sites are yet confirmed, we have begun to narrow down some possibilities. On this slide I have shown 3 peptides of interest for our project, snakin-1, BMAP18, and AHAMP1, along with their crystal structures. Below I have shown the potential locations or molecules of interaction that we are looking into. As of right now, these are our 3 primary simulations we would like to run once we have more information. That will be snakin-1 reacting with the damage repair enzymes for the cell membrane, BMAP18 with the cell membrane itself, and AHAMP1 with the phospholipids within the cell membrane. While creating our project design, one of our primary concerns was the use of genetically modified organisms for human consumption. With the controversy surrounding the topic, we wanted to gauge how realistic our project was to pursue. We met with some specialists and farmers in our community to see where they stood on the issue. The first interview we had was with Dr. Jonathan Nielsen, who is a researcher of potato health and plant stress physiology. In the interview, he mentioned ways around the GMO stigma, such as ensuring the inserted genes never end up directly in the tuber. When looking into preventing fusarium infections, we are also considering using topical application of our AMPs. This has changed the type of AMPs we are looking into to see which ones could effectively work outside of the potato as well. Our interview with David Hill was next, and it opened up our minds to see beyond just Fusarium infection. This shifted our design focus to look into nutraceuticals as well. Now we have begun looking into AMPs with specific health outcomes. One such AMP is BMAP18, which acts against the trypanosoma parasite that causes sleeping sickness. This shifted our focus to address the whole potato production line and see what other aspects could be improved. Next, we met with Florian Dieker, who is a potato provider for McCain and Frito-Lay. He talked about what makes potatoes vulnerable to Fusarium infection. This infection is caused by damage to the skin, which allows pathogens to enter. Since an aspect of preventing infection would be protection against membrane damage, we have begun looking into AMPs naturally found in potatoes for defense. The plant team has been doing work to see if we can increase the amount of these AMPs that occur naturally in potatoes. We have also begun a project where we want to trace the food line to see where potatoes end up for consumers. That way, we will see what the countries who receive these potato products think of GMOs and what kinds of regulations they have surrounding them. This will provide a bigger picture of global perspectives on GMOs, as well as the feasibility of our project. Our survey is made to include the thoughts and opinions of potato farmers within Canada. The survey will consist of 2 versions, which will be administered randomly by our chosen program. One version will include words such as GMO, genetically modified, and genetically engineered, and the other will not. This will help us to see the reasons why people are against GMOs, and the different ways we can address these reasons in our project for the upcoming year. We hope to better understand whether or not the stigma surrounding GMOs alone is the main opponent to their commercial use. This survey will allow us to see if the health benefits and decrease in crop loss that would result from our project are initiatives that are supported, and if the opposition stems solely from the use of genetic engineering. We'd like to thank the University of Lethbridge faculty, our PI, Dr. Angeliki Pantazzi, our advisors, team members, the University of Lethbridge Labs, our collaboration with the High School Lethbridge cGEM team, Dr. Mariana Ventrisi, Dr. Jonathan Nielsen, and David Hill, as well as all of our sponsors. On behalf of the Lethbridge cGEM 2020 team, thank you for listening to our presentation.